Okay, so since it is already 3 o'clock, I'll get started with this session. Um, I think no one is there in this session, but I'll still go ahead with the session. So, welcome everyone to week 5 of this TA session for the course Bioengineering and Interface with Biology and Medicine. The course code for this course is NOC 25 BT05, and I am Arjun. I am one of the TAs for this course for the period January to April for this 8-week course. So we are in week 5 right now, which is like the starting of the second half of the course. So the plan for this TA session will be the same. So I'll be going over a general recap of what happened in the lectures for week 5 of this course, following which I'll be having a short doubt session for everyone. After which we will be solving some problem statements from week 5 of this course so that you can take the answers and questions forward to solve some problems for the week 5 of your assignments. And then after that, we have already discussed 2 weeks of assignment answers in the previous uh, weeks of this TA session. So in this week, we will be going over week 3 answers for your assignment. So this will be the general plan. So I will start with this general recap. So in week 4, we saw this will be the TSL for week 5, but just as a recap for all the other weeks. In week 4, what we saw is in week 4 we saw an introduction to genetics. So we saw in general about Mendelian genetics and how he came up with the Mendelian laws of inheritance, which basically comprised of law of segregation and law of independent assortment in main. And after that, we saw how to solve Mendelian genetics problems by drawing Punnett squares as well as by looking at other charts as well. And while ending the week's lectures, we saw how there are other cases of non-Mendelian inheritance as well, where one of the dominant alleles can be not completely dominant. It can be it can show incomplete dominance as well as it can be co-dominant with any other dominant allele as well. So continuing from week 4, week five's initial lectures also dealt with genetics, and week five's lectures started with chromosomal basis of inheritance or chromosomal roots of inheritance, where we just went into a molecular basis of how the dominant and recessive alleles become part of chromosomes, and how any trait, any part of gene in the chromosome. So we already saw in week 4 that genes are compressed into nucleosomes using histone proteins, and these nucleosomes are compressed further to form this chromatids and chromosomes. So we saw how genes form part of this chromosomes, and we saw how deviations from any kind of chromosomes will be formed as a different kind of allele, and the crossing over between different kinds of alleles can lead to different types of progeny. And in this week, what we saw is just a extension of that, where we saw about where we started about chromosomal basis of inheritance, and we just saw a basic example of how the Mendelian genetics can happen, and how the traits corresponding to small experiments in Mendelian genetics, such as the round seeds and micral seeds versus yellow seeds and green seeds, how they can occupy different regions in the chromosome. And we also went to saw how a specific scientist called Morgan, Thomas Hunt Morgan, he investigated into this chromosomal based theory of inheritance using a fruit fly model, which is also called as a Drosophila. So he invented different kinds of experiments using this fruit fly model to see into how recombinant type of animals come into existence. So he invented new nomenclatures such as wild type as well as the mutant gene. So wild type is generally called as the um, so yeah. So the most commonly found character in nature will be labeled as the wild type, and any deviation from this wild type will be called as a mutant variant. So what the here they have a nomenclature for denoting wild type and mutant. So wild type can be denoted as W plus, and the mutant alleles will be denoted as W without any plus. And we also can see through Morgan's Drosophila based experiments that some genes which give rise to some characters are present on only one of the sex chromosomes. So every human being has 22 pairs of autosomes and one pair of sex chromosome. And we can see through Morgan's experiments that only some either the X chromosome or the Y chromosome only in most of the cases carries the traits for carries the genes for certain kinds of traits. And here we can see for one such example where a red-eyed male is crossed with the white-eyed female, and the red eye is claimed to be as a wild type variant, and the white eye is claimed to be as a mutant variant. So the red eye is denoted by W plus, which is on the X chromosome and not on the Y chromosome, and the white-eyed female mutant is denoted by W, which is on the X chromosome. So when we cross the wild type with the when you cross this wild type with this mutant, we can get these kinds of progeny, out of which one is a female wild type, one is the uh, mutant male, and one is a mutant male here, and one is the female wild type. So we can see a specific trait here. So the white-eyed mutant variant, although it's a since it is a recessive trait, uh, when it appears on only one of the chromosomes, it still remains as a carrier and not as a trait in the female Drosophila. But on the male Drosophila, since there is only one X chromosome, and since that mutant allele happens to be on that X chromosome, it is followed the male progeny, which is arising out of this cross, comes to be as having white eyes. So this is what Morgan came to have discovered as a chromosomal that genes which are on the chromosomes tend to have different properties, and the crosses between different variants, the wild type and the mutants, give rise to different kinds of progeny. And in the first lecture, we also saw, yeah, so this, I also explained that this wild type and mutant gene, and some of these genes are sex linked. So they can be present on only one of the chromosomes and not on the other. And uh, when some of the crosses happen, the sometimes what can happen is when not all of the chromosomes are affected, then the resulting individual can be a carrier of the condition and not a affected and cannot be affected by the condition totally. And we will be seeing how some such disorders such as hemophilia can be expressed in the whole of a family, in wherein some of the people will be carriers and some will be affected. And we also saw an important fact about this inheritance, that if a specific condition is possessed by fathers, fathers can pass on this condition only to daughters, and if any specific mutant condition is possessed by these mothers, mothers can pass on to both sons and daughters. So using these facts, when we solve problems, we'll be using these facts to solve it. And later, we also, in the subsequent lectures, we also saw what is like about linkage as well as by chromosomal disorders. So in these lectures, we saw we were introduced to a concept called as recombinance and recombination. So recombinants are organisms which are like which have a character which is different from the parental type. So parents will have a specific type of combination of alleles. If the progeny has any different combination of alleles apart from the parent, they are claimed to be known as recombinants. And the process of recombination is once again. Yeah, and the process of recombination is just to said to be exchange of this nucleotides. So chromosomes from both the father and the mother come together to form this progeny, and while coming together, they cross over with each other. Where we can see here, this is called this crossing over, and this is the site of crossover. So both the father and the mother have a different homologous chromosomes, and as a result of this uh, crossing over, so nucleotides from this chromosome can go to switch over to this chromosome and vice versa. So as a result, we get this recombinant genetic information, which results in the production of recombinants. So, and we also came to know about the fact that this recombination happens in the prophase one of meiosis between homologous chromosomes. And furthermore, we also saw that how this recombination is useful, how this recombination can give to um, positive traits as well as negative traits. So initially in plants, we saw how this recombination can give to positive traits, and we also saw how important it is to perform a process called as karyotyping. So karyotyping is a process where we come to know the entire set of chromosomal information from any progeny or from parent. This is to study any form of deviation to find out if the resulting progeny will have any diseases or disorders. And in the subsequent part of the lecture, we also saw about chromosomal aberrations, where we saw that the information present in the chromosomes can be um, added, deleted, inverted, or like changed over to some other place, and this will result in structural and functional changes to the resulting progeny. And we also saw how this kind of modifications to the genetic material can lead to some disorders. We mainly saw uh, disorders which whose bases are based on the trisomy. That is, instead of two chromosomes, we have an additional chromosome. So trisomy of different chromosome number can lead to different diseases. So trisomy of the 21st chromosome can lead to Down syndrome. Trisomy of 18th chromosome can lead to your Edwards syndrome, and trisomy of your 13th chromosome can lead to Pateau syndrome. And we also saw about this Turner syndrome, which is a non-trisomy disorder. And that's all about this linkage in chromosomal disorders. After that, we took a short deviation from genetics. Like, it is also a genetic thing, but we took a short deviation from how genetics works to study what is the basis of genetics about DNA, RNA, and protein. So we know that there are three fundamental molecules which are involved in this transmission of genetic information, which is the DNA, RNA, and protein. And we saw how classical genetic experiments led to the discovery of how DNA is the genetic material in humans as mammals as well. So the course instructor Dr. Sanjeeva Srivastava, he discussed about this Griffith experiment, where different types of mice, the same uh, different types of mice were injected with different strains of Staphylococcus aureus causing pneumonia, and wherein the mice were exposed to different conditions. So when Staph aureus having a rough strain, which is a non-virulent strain, were injected into the mice, mice lived. Whereas if the Staph aureus with a smooth strain was injected into the mice died. And they observed that when the uh, smooth when the heat killed smooth strain and the rough strain were injected simultaneously into the mice died. So Griffith experiment, Griffith was somehow able to explain that some of the, even though the smooth strain was heat killed and administered, some of the genetic information from the heat smooth strain where was able to be passed on to the rough strain, which was able, and the rough strain staff warriors was able to replicate the smooth strain virulency, which led to the death of the mice. So Griffith concluded that some sort of genetic information is being passed on to the next generation, which aids in the replication of that genetic material. But they were still confused about what kind of genetic material can be, whether it can be DNA or protein. So after that, Hershey and Chase conducted an experiment using bacteriophages where they labeled where they had two conditions where they labeled the phosphorus present in the DNA, and where in the other case they labeled the methionine present in the proteins. So they had two cases where they had labeled phosphorus and where they labeled methionine, and then they the bacteriophages were made to infect bacteria and kill them, so that and they observed which kind of material is being passed on. So if protein were really the genetic material, in the progeny, in the successful progeny also, they should be able to observe the radioactive methionine, which they weren't able to observe. But when they um, when they administered the bacteriophage labeled with whose DNA was labeled, they were able to observe that in the cells which were affected with which were infected with bacteriophages, the DNA was having radio labeled phosphorus. So through this experiment, they were able to conclude that DNA is the genetic material that is being passed on from generation to generation. And there were a lot of, after concluding that DNA is the genetic material, there was a lot of confusion about how the DNA is being replicated. So people had a lot of theories about it. They had a, semi-conservative model, they had a conservative model, and they had a mixed model where different theories propose about different models. And semi-conservative model came to be the most widely renowned widely accepted theory for the DNA replication, and this came to be a confirmation through this experiment by Messelson and Stahl. So what they did was they radio labeled both the strands of DNA using N15, that is radioisotope of 15 nitrogen, and what they did was they allowed the DNA to replicate after several cycles. So after one cycle, they isolated both the DNA and they performed density gradient centrifugation, where they were able to observe a different, um, where they were able to observe only one band of DNA, which corresponded to the N14 N15 hybrid, where one strand was normal N14 and one strand was N15 labeled. When they allowed both of these strands to replicate once more, and after that, when they isolated the samples and they performed density gradient based centrifugation, they were able to observe that they were able to observe two different bands in the density gradient based centrifugation, out of which one band was purely N14 N14, where both the strands were N14 labeled with N14 nitrogen, but and they and another set of strands had N14 N15, where one strand had N14 DNA and the other strand had N15 DNA. And coincidentally, the proportion of this N14 N14 to N14 N15 hybrid was 1 is to 1. So through this experiment, they were able to confirm and accept the semi-conservative model of DNA. And in the final set of lectures, the course instructor focused on explaining about bacteria and viruses, where he first started with bacteria, and most of the lecture was about bacteria, where he explained about different shapes and sizes of bacteria, and how they were able to move. Like bacteria had different mechanisms for moving, and like chemotaxis and urotaxis, and bacteria mainly move by chemotaxis, by chemical gradient signaling. And he also explained about how bacteria used to reproduce. Bacteria reproduce asexually through mostly through binary fission. And he also explained how the bacteria were broadly divided into two different categories, gram positive and gram negative bacteria. And there was a demo on how to perform this gram staining as well, where gram staining usually involved this administration of crystal violet stain and iodine, and after which we wash it with alcohol and we put a counter strain called safranin. So gram positive bacteria will appear purple on a, will accept this crystal violet stain. So crystal violet stain targets the peptidoglycan covering. So gram positive bacteria have a very thick layer of peptidoglycan on their surface. So this crystal violet targets the peptidoglycan layer, which is being accepted by the peptidoglycan, and the peptidoglycan after staining purple, it gives out this color. Gram negative bacteria, on the other hand, it doesn't have much of peptidoglycan. It has a lot of other membrane proteins. So those proteins accept this saffron stain and they stain red. So gram positive after the gram staining, gram positive bacteria appear purple in color, whereas gram negative bacteria appear red in color. And we also saw about how the different uses as well as the different diseases that can be caused by bacteria. Bacteria had like different kinds of diseases such as like anthrax, typhoid, and pneumonia. But on the other hand, it can also be bacteria can also be used in a lot of useful purposes such as recycling of organic waste, decomposition, and as well as for nitrogen fixation. And we also saw about how bacteria can have a symbiotic relationship with other types of organisms in a beneficial manner. And after that, finally, for a short part of the lecture, we saw more about viruses and how they are like and how they are like a class apart from all the living organisms, because they are actually not living outside, but they are only classified as being living when they are inside a host system. And we saw the different types of viruses such as the bacteriophage as well as the influenza virus, and we saw a common structure where the genetic material is enclosed in a protein code called the capsid, and in most of the viruses. And after that, we saw about lot of viral based diseases, which included poliovirus, SARS-CoV virus, as well as your swine flu virus. Swine flu virus came under the influenza kind of viruses. So we also concluded the lecture using classification of influenza, how the different types of how the different types of influenza are like once again. Just a second. Yeah. So we saw how different types of influenza can be classified into H1N1, H1N2 based on the two types of proteins. So the H and N represent the hemagglutinin and neuraminidase, and depending on the type of hemagglutinin and neuraminidase present on the viral capsid coat, we are able to classify the influenza virus. So that's all about the weekly recap for this session. So I'll leave the room till 3:20, 3:30 for a short doubt session, following which we will continue discussing about some problem statements for this week. Okay, so since no one's in the room right now, I'll proceed with the discussion of problem statements. So I'll upload this lecture to YouTube as well, and if you guys have any doubts there, you can directly post it in the comment section, or you can ask those doubts like that, or the next doubt, or you can ask those doubts in the next TA session. So consider the given family. So there is a picture here of a family where a male is a phenotypically normal person and the female is also a phenotypically normal person, and they have like five progeny. Phenotypically, three are phenotypically normal and two are diseased. So what could be the most possible inheritance pattern of this disease? So they are asking what could be the like common this thing. So they have four options. One is autosomal dominant, one is autosomal recessive, and then X-linked dominant, and then X-linked. Third option is the Y-linked dominant, and X-linked dominant. So the answer for this will be autosomal recessive, because if it's a sex linked so how to go around this question is, so first we'll confirm whether this is a autosomal disease or a sex linked disease. So if it is a sex linked disease, and yeah, so if it is a sex linked disease, one of them should be a at least be a carrier of the disease, for the diseased carrier of the disease through the sex chromosome, for the children to be diseased. But that's not the case. Both of them are phenotypically normal, so it will not be a sex linked disease. And if it is a autosomal disease, look at the proportion. So out of five people, three people are normal and two people are diseased. So the proportion of the disease to the normal is very less. So it's a autosomal recessive disease. So I'll go on to the next question. So color blindness is a X-linked recessive disorder. What will be the chance of having a colorblind male child to this couple? So one is a colorblind. One is a colorblind grandfather for this child. Uh, The child has a colorblind grandfather and a colorblind carrier grandmother. So um, yeah, so colorblind grandfather and colorblind carrier grandmother, uh, who give rise to a phenotypically normal father, and the phenotypically normal father will have a colorblind carrier mother. So, what is the proportion? Uh, what is the chance of having a colorblind male child to this couple? So this kind of problem is pretty simple. So if it's a colorblind carrier mother, so colorblindness is a recessive disorder. So we'll carry on from this itself. So so the colorblind carrier mother will have, I'll denote colorblindness using this C variant. So if it's a colorblind carrier mother and if colorblindness is a recessive is a X-linked recessive disorder. So only if both the X's are having this colorblindness gene, then the mother will be colorblind. But the mother is a colorblind carrier. So only one X chromosome will be a colorblind will respond to the colorblindness character. So mother will be having a XC X normal character, and we have a phenotypically normal father. So we are, and we also know that X-linked recessive disorder in a male, it needs to have only one X chromosome. A father will have XY. So if he actually has color blindness, he will have XC. but he is normal. So his X chromosome will also be normal. So we have a XY and then we have a XCX. So, and they are asking about what will be the chance of having a colorblind male child. So if it's a male child, this Y chromosome has to cross over with any of this X chromosomes. So if it's a male child, the only possibilities are XCY and XY. So this XY will give rise to a normal male child, whereas XCY will give rise to a colorblind male child. So the chance of having a colorblind male child is 50%, which is option C. So I'll kind of again explain how this 50% came to be existent. So forget about this colorblind grandfather and colorblind grandmother for now. So we have a phenotypically normal father and we have a colorblind carrier mother. And we know that color blindness is a X-linked recessive disorder. So if it's a recessive disorder, uh, both the X chromosomes of the mother needs to be having this color blindness trait for the mother to exhibit the color blindness. And if any one of the X chromosomes are exhibiting the color blindness character, then the mother will be a carrier. So the colorblind carrier mother's karyotype will be XCX. And on the other hand, the phenotypically normal father, we'll take the case of the phenotypically normal father. The father should, if he is having color blindness, he will have XCY because it's a X-linked disorder. So the Y won't have the color blindness character. So since he is a normal father, he will have a karyotype of XY, where the X is a normal X chromosome and it's not a mutant for color blindness. And they are going to cross over to give a male child. So in case of the male child, the X chromosome from the father will not be involved. So we only have to consider this Y and Y crossing over with XC and X, which will either give rise to XCY, which is a colorblind male, or a XY, which is a normal male. So there are only two options here, colorblind male and normal male. So the chance of having a colorblind male is 1 by 2, which is 50%. So I can come back to this question whenever anyone has doubts. So I'll go on to the next question. So consider linked genes between body color and eye color on a fly. So we also saw about linked genes on a chromosome. So linked genes are those kind of genes which are like present on the same chromosome but with a distance from each other. And the farther the linked genes are, the higher the chance of recombination from them. So the statement is the golden bodied red-eyed are normal ones. I'll just switch to the pointer. So the golden bodied red-eyed are normal ones, whereas the grey bodied grey color-eyed are double mutant ones. So what is the phenotype with which the female fly was crossed to produce the four offspring? So they are given a cross here, and so G plus are the golden color, and R plus are the red color, and G as the gray color, and the R is the um, black gray eye color. Yeah. So both of them are crossed together to give the G plus R plus, which is once again the golden, which is once again the golden body, the red-eyed color. So they are asking which should be crossed over to give this kind of combinations. I'll give the room like 2 3 minutes to think about it. Then I'll go again. actually a pretty simple concept if we think about. Uh, we discussed about we have been discussing about one specific concept in Mendelian genetics over the past 2 weeks. So if we kind of look about that, like look to this problem through that, then we can easily solve this problem. Okay, I'll go on to explain the answer. So that Yeah, I'll go on to explain the answer. The answer is this, which is the gray-bodied, gray-eyed color. So look at this cross in particular. Yeah, one second. So, yeah, look at this cross in particular. So this is a hybrid phenotype which is having one dominant and one recessive and one dominant and one recessive of both the body color as well as the eye color. And when it is crossed with something else, it is giving you a phenotype of all the four possible phenotypes in 1 is to 1 is to 1 is to 1 ratio. So what we saw about a concept called as a test cross, which is a cross of cross between a hybrid phenotype as well as a recessive phenotype in individual to produce phenotypes of all the possible phenotypes in 1 is to 1 is to 1 is to 1 ratio. So, for to a test cross to be performed, you need to cross it with a completely recessive, like traded in the individual. So, which is the gray-bodied, gray-eyed color individual. So, this is the gray-bodied, gray-eyed individual. All the four individuals do not correspond to a test cross, and they will not give rise to this phenotype difference. I'll like give I'll give like 2 3 minutes to like uh, go through this question again, and I'll go on to the next question. So basically, for this question, you need to remember what a test cross and like in which cases do we use test cross and back cross, and under a test cross and a back cross, what kind of genotypic and phenotypic ratios will we be obtaining? And this is a classic example of a test clause, which will give you 1 is to 1 is to 1 is to 1. Okay, so I'll move to the next question. I think that this question is clear. So, one second. Oh yeah, so three questions are done. So, fourth question is kind of simple. So, we saw a theoretical question. Which of the following syndrome differ in terms of chromosomal aberration from the other ones? So, all four options given here are like chromosome aberrations only. but one of them is like kind of an odd one out. We just have to find out which one. The options given here are Turner syndrome, Edwards syndrome, Cutoff syndrome, and Down syndrome. So even in the recap, we saw that Down syndrome, Cutoff syndrome, and Edwards syndrome, all of them are chromosomal disorders associated with trisomies of different chromosomes, chromosomal 18, 21, and 13. Whereas Turner syndrome is not a Turner syndrome is not a chromosomal aberration based on trisomy. It's different. It's based on a X-linked genetic difference. So the answer is Turner syndrome. So I'll go to the next question since it's a very theoretical question. So suppose Ramesh is studying the following chromosome. Arrange the location of the genes on the chromosome based on the following recombination frequencies. So this is the following chromosome that they are given, and they are given different kinds of genes with different letters: alpha, beta, gamma, delta, epsilon, and omega. I think it's omega, or now I consider it omega. I forgot what it is. So arrange the location of the genes on the chromosome based on the following recombination frequencies. So they have given the recombination frequencies of each one of these genes, and yeah, so each one of them should match. Each one of them should match with each other. So alpha beta are separated by 20 map units, and this I think it's omega beta are separated by 13 map units, and so on and so forth. So all of these are separated by certain map units. Map units are distance of the chromosome, and the farther the map units are, the larger the recombination frequency will be. This is the basic concept. So you just have to do the math around here and figure out which option makes more sense here. I mean, the shorter way is to just put in one value here. So if they're given beta epsilon, and they you just have to calculate we just have to say beta epsilon, and then just figure out if all of them make sense. If epsilon delta is making sense, gamma delta is making sense, and all those stuff. So I'll give like 5-10 minutes to go through this problem and get the answer. If not, I'll after that I'll start solving it. And in most of the cases, not all information will be available as well. So you just have to ignore some of the some of the information and try to work around that. See, so how come? If let's just try to solve C in that case then. So Unity is saying C. So C is saying the sequence will be the correct answer. But let's look at the sequence. So alpha epsilon will be 5 ampere. I'll just write it up with a pen as well. So alpha epsilon they are saying 5 map units, and then epsilon delta they have epsilon delta they are given 15 map units, and delta gamma is giving 27 map units, and gamma to beta they're giving 13 map units. So so from alpha to beta technically it should be 5 plus 15 20, 20 plus 27 47, and 60. So alpha to beta technically should be 60 map units, but here they are given alpha to beta is only 20 map units. So in that case your uh, option C will not be a correct answer, because the map units prescribed here using the other options exceed the actual map units from alpha to beta, which is like 60. You can maybe try the same option for the other answers as well. So here beta to epsilon is not given. Epsilon to delta is given 15, and delta to omega is given kind of 27. So you can just see whether epsilon to delta or epsilon to gamma is given somewhere here. No, that's also not given. So gamma to delta is given. So gamma to delta is given 8. Gamma to omega is given 8 map units, and gamma to alpha it's not given. So actually he this also makes sense. So gamma to delta. So yeah, gamma to omega is 8 map units, but here omega to alpha is given 7 map units, which is technically not possible. Then alpha has to come before gamma, which is not the case here. So this can't be the correct answer. And C already we solved it. It's not the correct answer. So in this case, so gamma to epsilon, they haven't given here, and epsilon to delta they haven't given here. They are given only epsilon to alpha, which is like, oh yeah, so simple. So epsilon to alpha they are given 5 map units, but epsilon to delta they are given 15 map units. So this is 15, but this is 5, which is not possible. So this also can't be the correct answer. So this should be technically the correct answer, but we'll just see how it works out. So gamma to delta they given 15 map units, and they haven't given any other this to this. So delta to omega is 27 map units. Yeah. Epsilon to alpha they are given 5 map units, and alpha to omega they are given 7 map units. So here it matches. So 15 plus 5 plus 7, which gives you 27, and already delta to omega gives you 27 map units. And we can also if you can also see other this thing. So your omega to gamma gives you like 8 map units, and omega to beta gives you 13 map units. And so technically your gamma to beta should give you 5 map units, but it's not given here anyways. So it can be possible that it's 5 map units, but we can also like kind of make it that. So overall we can see that all of this information comes together properly, and I think we are just missing out on like one thing, which is alpha to beta. So yeah, so alpha to beta also they are saying that it's around like 20 map units, and if we make through this information 7 plus 8 and 20, your gamma to beta should be 5, which is also the same case here because 8 plus 5 gives you 13. So all the information given in this question matches with this answer. So your correct answer will be this option. Option D. So I'll just give a short break for everyone else to go through this answer as well. If in case you are watching this recording in YouTube, you can also pause this lecture and you can uh, try to solve the problem yourself as well. So I'll start the next question from 350. In the actual exam, these kind of questions might consume a lot of time as well. So it would be good if you are like if you can quickly calculate the map size between these kind of options in the exam. So I'll go on to the next question. So question number 6. Read the given statements carefully. So it's a entirely theoretical question. Statement 1 is all the genes are essentially present on the chromosome, okay, which is like a true statement. Statement 2, genes that are located on the same chromosome will always pass together from one generation to another, which is not true because we also saw about the concept of crossing over and recombination as well. Statement 3, the distance between two genes can never be mapped without cloning experiments, which is kind of true. And statement 4, all genes are essentially overlapping present on the chromosome, which is also true. So, depending on the reading frame, your genes can your genes can be overlapped, and the specific gene can be expressed depending on the reading frame. So pick the incorrect interpretation for the molecular explanation of chromosomal theory of inheritance. So out of this, all these four statements, we found only one of this statement to be like false, which is that genes located on the same chromosome will always pass together because they can also be recombined and then passed on to generations. So the incorrect statement here is statement 2. So I'll go on to the next question. So in an experiment, two genes were assorted independently. Which of the following points explain the best about the chromosomal location of these two genes? So it's just saying that two genes were assorted independently. Which of the following points explain the best about the chromosomal location of these two genes? So the options are they are located in close proximity on the same chromosome. Crossing over between the genes does not occur. The genes are either located on different chromosomes or are far apart on the same chromosome, and the location of the genes cannot be predicted based on the given data. It's a entirely like a logical question. So the answer is the genes are like they are either located on the different chromosome or far apart in the same chromosome because they are being assorted independently. So I'll go on to the next question. E. coli, Escherichia coli, fully labeled with 15 nitrogen. So 15N is the radioactive isotope, is allowed to grow in 14N medium. 14 nitrogen is the normal non-radioactive medium, which is the constituent of DNA. So, pick the correct statement about the two strands of DNA molecule of the first generation bacteria. So, they are having a 14 nitrogen with a medium with 14 nitrogen medium, and they are having E. coli which is growing on that medium which is fully labeled with 15N. So we also saw in the Messelson and Starr experiment that it will 15N medium will semi-conservatively they will replicate, and each of the complementary strand will be of 14N medium. So the options given here are DNA strands of daughter cells will have different density and they do not resemble parent DNA strands. DNA strands of daughter cells will have same density and they resemble parent DNA. DNA strands of daughter cells will have different density but resemble parent DNA. DNA strands of daughter cells will have same density but do not resemble parent DNA. Out of which the fourth option is correct. They will have like different third option is correct. That is, they will have a different density compared to the parent DNA. Don't get confused using this because while reading I got confused. So they will have a different density compared to parent DNA. So that because the parent DNA will have 15 and 15 in density, so they will have a higher density. But the daughter strands will have 15 and 14N. So one of the strands will be 15N labeled and one of the strands will be 14N. So they will have a different density compared to the parental DNA, but they will not like they will resemble parent DNA because they are having the same constituents as well. So I'll go to the next question. So a pointed frontal hairline is popularly referred to as widow's peak. So a lot of people have widow's peak as we can see outside in a lot of with a lot of people. So this is an example of widow's peak where there is a pointed hairline and it is inherited as a autosomal dominant trait. So consider the following situation. So widow's peak, a male with widow's peak is crossed with a woman with straight hairline to give a progeny with straight hairline. I actually forgot it. I don't know if I have not got the same question. It's okay. I think the question is what is the probability of getting a woman with female child with straight hairline, which is 50%. I'll edit the question. I'll try to edit this question and upload this PPT in while I'm uploading the final PPT as well as the video. But for now, it's not. The question is not complete. So last question, and this is a very pretty simple question. So choose the correct option in the context of gram staining. So we saw about gram staining in the first we saw about gram staining in the recap of the lecture as well as in the lectures. So gram staining has a primary stain, it has a destaining part, and it has a counter stain part. We have to find out which one is what. So this is entirely theoretical. We know that primary stain is crystal violet, the destainer is alcohol, and then the counter stain is saffron, which is option D. So the option is primary stain crystal violet, destainer alcohol, and counter stain as saffron. So that's all I have for the problem statements. I'll go again through all one of them to just this thing. So question one: consider the given family. What could be the most possible inheritance pattern of this disease? So this is autosomal recessive disease. Color blindness is an X-linked recessive disorder. What will be the chance of having a colorblind male child to this couple? The answer is 50%. Consider linked genes between body color and eye color of a fly. So golden body red eye and then normal ones, and grey body grey color eye are double mutant ones. So what is the phenotype with which the female fly was crossed to produce the four offspring? So this is an example of S cross. So the answer is this. The male is grey body, grey eye color. Which of the following syndrome differ in terms of chromosomal aberration from the other ones? Answer is Turner syndrome. Suppose Ramesh is studying the following chromosome. Arrange the location of the genes on the chromosome based on the following recombination frequencies. We did some math and we found out that this set of chromosomal mapping is the correct location of the genes. And this is a four statements where we found out where they are asking us to pick the incorrect interpretation of molecular explanation. So among all the statements, statement 2 is the incorrect one, which says that genes are located on the same chromosome will always pass together from one generation to another. And seventh question, in an experiment, two genes were assorted independently. Which of the following points explain the best about the chromosomal locations of these two genes? And the answer is the genes are either located on different chromosomes or are far apart on the same chromosome. Question number 8 is Escherichia coli, fully labeled with 15N, is allowed to grow in 14N medium. Pick the correct statement about the two strands of DNA molecule of the first generation bacteria. And the answer is option 3. DNA strands will have different density but resemble parent DNA. Question number 9, we don't know, we'll just skip it for now. Question number 10 is the correct option about the primary stain, counter stain, as well as the D stainer and gram staining. The correct answer is primary stain is crystal violet, D stainer is alcohol, and counter stain is saffron. So that's all for this problem solving statements of this week. So I'll have a short doubt session for like 10-15 minutes. So around 4:10 or 4:15 we'll resume where we can discuss about the your assignment answers for week 3. So I've just compiled the answers for week 3, and week 3 is like answers were mostly about Sankar sequencing and all those stuff. So we'll just discuss those answers and we'll just see if there are any probable doubts that arise out of that questions, and then we conclude the lecture. TA session. Sorry. Okay, so we will move on to discussing the questions and answers for like the assignment answers for the week 3 session. So, which are the following statements? Question number one. Which are the following statements are incorrect about Sanger sequencing? So they are given four options and we have to choose which one of these are incorrect. So DDNTP terminates growing DNA strand because it lacks 3-hydroxyl OH group, which is a correct statement. Synthesis of new strand starts at 5' end, which is incorrect because it's like this thing. Fluorescent detector senses the color of each fluorescent tag, which is true. DNA template, primers, polymerase, DNTPs, DDNTPs are required as reagents for performing Sanger sequencing, which is true. So there is only one incorrect option here. Synthesis of new strand starts at 5' end. So there's the incorrect statement is the correct answer, which is this. So next question. So a food technologist wants to compare the gene expression difference of yeast of Kashmir and Kanyakumari. How should he design the experiment using microarrays? So I'll read the question once again. A food technologist wants to compare the gene expression difference of yeast of Kashmir and Kanyakumari. How should he design the experiment using microarrays? So there are four options. He can extract total RNA from the samples and label and hybridize on the slide. He can extract total DNA from the samples and label and hybridize on the slide. He can spot cDNA on the array, extract total RNA from the samples, label and hybridize on the slide. He can spot either of DNA or RNA on the slide. So the answer for this is spotting cDNA on the array, extracting total RNA from the samples, labeling and hybridizing on the slide, which is the way for doing microarray based experiments. So whichever RNA fits with the complementary DNA of the sample, it will hybridize and it will fluoresce, which giving you the output or the genetic expression difference. So question number three, uh, Nikhil works in a pharmaceutical company and wants to launch a new therapeutic protein for which he intends to manufacture that protein on a large scale. He thought to purify the protein using affinity chromatography. He asked the technician to use N-terminal histag for better protein purification. So, which we already saw in week 3 that we use this N-terminal histidine tag using affinity chromatography for isolation of proteins and for protein purification. So, what is the most important factor to consider while choosing the correct host for expression of the recombinant protein? Length of gene, gaps between exons and introns, presence of transposons, and post-translation modifications. So, out of this and four options, post-translational modifications seem to be the most correct answer for this. So the answer, because we are focusing on this protein purification, so the correct answer will be post-translation modifications. So next question, which of the following enzymes is used to synthesize a cDNA library but not to synthesize a genomic library? So options are restriction enzymes, DNA ligase, reverse transcriptase, and DNA polymerase. And the answer is like pretty theoretical. Reverse transcriptase. So that is used to synthesize a cDNA library from a RNA sample, but it's not used to synthesize a genomic library. So question number 5. Below are the two statements labeled as assertion and reason regarding the production of golden rays. choose the correct option. So they are given an assertion and reason. So we have to see if, sorry, so we have to choose whether the assertion and reason are true and whether the reason is the correct or the wrong explanation for the assertion. So the assertion is that eating golden rice will prevent vitamin A deficiencies that cause blindness and immune system problems. So golden rice, as we know, it's a, and reason is it produces beta carotene, which is a precursor to vitamin A. So both the assertion and reasoning are correct, and the reason is the correct explanation for the assertion, because golden rice has beta carotene, and having beta carotene will prevent vitamin. Beta carotene is a precursor to vitamin A. So having golden rice having, which is having beta carotene, which will prevent vitamin A deficiency because your vitamin A production is becoming higher. So that will prevent you from having diseases like night blindness and immune system problems. So the correct answer is both the assertion and reason are true, and reason is the correct explanation for the answer assertion. So far, all the questions have just been theoretical. So next question is, transgenic plant cassava is said to have the ability to fight world hunger. Once again. Yeah. Transgenic plant cassava is said to have the ability to fight world hunger. This is so as it has been made to produce iron and beta carotene. Which of the following can be used as technique to insert the recombinant plasmids with the iron and beta carotene gene producing, sorry, iron and beta carotene producing genes into the cassava plant cells? So they have some they have cassava, and so it is made to produce iron and beta carotene. So using iron gene and beta carotene gene, it is made to fight world hunger. So using what technique can you insert the iron and beta carotene recombinant genes into the recombinant plasmid? So look at the options here. So electroporation, electrophoresis, polymerase chain reaction, and chromatography. Chromatography is used for isolation. It has no use in insertion. Polymerase chain reaction is in use in amplification, but unless you insert, you can't amplify. Electrophoresis is you same as like same principle as chromatography, but it is just used to for purification. So the only correct option here is electroporation, which is using the electrical field to disturb the plasmid so that your insertion can happen. So read the given statements carefully regarding cDNA synthesis. Seventh question. So RNA's first statement is RNA is zapped to be avoided during RNA extraction as it promotes RNA hydrolysis. So RNA is zapped to be avoided during RNA extraction as it promotes RNA hydrolysis. DNA's enzyme need to be added during DNA. Sorry. DNA's enzyme needs to be added during RNA extraction to prevent DNA contamination. Phenol and guanidine isothiocyanate are responsible for the separation of RNA from DNA and protein. So I'll read statement 3 again. Phenol and guanidine sorry, once again. So phenol and guanidine isothiocyanate are responsible for the separation of RNA from DNA and protein. And statement 4 is chloroform is added to lyse the cells and bring out the contents. Which of the following statement is correct? So out of all these 4 statements, only option 2 seems to be correct. DNA's enzyme needs to be added during RNA extraction to prevent DNA contamination. So for cDNA synthesis, we'll first extract the RNA sample. We'll isolate the RNA from the sample of interest, and for doing RNA extraction to get the pure RNA, you have to add the DNA's enzyme so that it lyses the DNA which is inside the sample, so that the DNA doesn't come along with the RNA and preventing DNA contamination. So the correct option here is option 2. So, question number 8. Rohan has purified a recombinant protein. He wants to check his molecular weight. Which are the following methods he could use to check the molecular weight? So, options given are SDS page electrophoresis, mass spectrometry, analytical size exclusion chromatography, and all of the above mentioned techniques can be used. And the correct option here I will say is all the above options, because all of them are like all of them can be used to check the molecular weight of any protein. So all of the above mentioned techniques. Question number 9 is Suresh has purified a N-terminal his-tagged recombinant protein by affinity chromatography. However, SDS page showed bands in addition to the target protein. So, how might Suresh can improve this purification step? So, I'll read the question once again. Suresh has purified N-terminal his tagged recombinant protein by affinity chromatography. However, SDS page showed bands in addition to the target protein. How might Suresh can improve this purification step? So the correct answer for this question is to use protease inhibitors, so that if you use the proper protease inhibitor, it will lyse the appropriate protein for it. Thereby, you can isolate your his tagged recombinant protein more efficiently. Therefore, you will get only one band in your SDS page. And the last question, like so far, all the 9 questions have been like only theoretical only, so it's pretty much easy to solve. So only the last question is kind of mathematical. So one second, just give me 1 second. Sorry for the disturbance. I got a call. So this thing. So yeah. So far all the 9 questions have been like theoretical only. Only one of this question is like problem. So we'll try to solve the problem as well actually. So question is, Nikita has extracted RNA from certain yeast cells. She wants to quantify it for further downstream experiments. She dissolved the extracted RNA pellet in 100 microliter of distilled water. She diluted the sample in distilled water in the ratio of 1:50. The absorbance at 260 nanometer came to be 0.46. What is her total yield of RNA in microgram? So I'll read the question once again. Nikita has extracted RNA from certain yeast cells. She wants to quantify it for further downstream experiments. So RNA and quantification of RNA. She dissolved the extracted RNA pellet. So she has some total yield of RNA in some microgram. She has dissolved this RNA pellet in 100 microliter of distilled water, and then she has diluted the sample in again distilled water in the ratio of 1:50. So two times dilution. So 1 and 100 microliter of water, and once again in distilled water in the ratio of 1:50. And after two times dilution, the absorbance has come to be 0.46. So what is the total yield of RNA in microgram? That is the question here. So Just to solve the question, I'll just give um, a hint. So typically in RNA based as RNA quantification based experiments, we take absorbance at 260. So if our absorbance at 260 is equal to 1, so here it's 0.46, right? I'll write it in detail when we have, I don't know whether I have like, it's okay, I'll write it using this itself. So, um, so if, yeah, so if absorbance is equal to 1, then it means that the amount of RNA present in the sample is 40 microgram per ml. So the if the RNA the final sample, so final sample, if it is if the RNA is 40 microgram per ml, then you will get absorbance as 1. That is like a fixed prefixed fact kind of stuff. So if absorbance is 1 and gives you if 40 microgram per ml gives you 1, how much will 0.46 absorbance will give you? You can do a small calculation with respect to that, and you will get around 18.2 microgram per ml. So I hope everyone, if you're watching this recorded video on YouTube, you are getting this. So your absorbance of 1 means that there are 40 microgram per ml of RNA, which is also like a salt in this lectures as well. And so if you have 0.46 of absorbance, you will be having 18.2. So 0.46 into 40 divided by 1, which gives you 18.2 microgram per ml. So now what we have to do is, so this we work this up in reverse order to find the total yield of RNA. So after, so the last step of dilution, they have diluted it to 1:50. So dilution 1, so let's just say as D1. So it's 50 times dilution. So you multiply 18.2 times 50, which gives you around um, 91. Yeah. Oh, sorry. 0.46 into 40 is 18.4, right? Sorry. Yeah. Sorry, I'm very sorry for this. Sorry. So 18.4 microgram per ml, and dilution is 18 point. So we are diluting it by 50, 1:50. So before the dilution, it will be 18.4 into 50, which is 920 microgram per ml, and before 920 microgram per ml. So ml, and then first step is she dissolved the extracted RNA pellet in 100 microliter of distilled water. So the final yield will be 920 microgram per ml into 100 microliter is like 0.1 ml. So you convert it into ml so that your SI units will get cancelled. So your ml and ml get cancelled. So 920 into 0.1 is 92 microgram. So your correct answer will be 92 microgram. So I'll go through how to solve this problem once again. So we know that the final whatever we know that whatever RNA we have after dilution in 100 microliter of distilled water and after diluting it further in 1 is to 50 ratio, we are getting an absorbance of 0.46. And we know that 40 microgram per ml of this kind of RNA will give you an absorbance of 1. So using this we have to find out how much amount of RNA in microgram per ml will give you absorbance of 0.46, which upon calculation is giving us 18.4 microgram per ml. And this 18.4 microgram per ml, if we hadn't diluted it by 1 is to 50, then it will be having the quantity of 18.4 into 50, which is 920 microgram per ml. And after this, they, if this 920 microgram per ml, if it hadn't been dissolved in 100 microliter of distilled water, that is 0.1 ml of distilled water, then it would have been 920 microgram per ml into 0.1 ml, which is like 92 microgram, which is the correct answer. So I hope this you guys get how to solve this thing. And that's all the questions there were there for this week 3 of assignment. So I'll keep the room open till like for another 10-15 minutes for doubt session. And I'd like to make one more important announcement regarding next week's TA session. So I think I have already informed this to NPTEL team, but still I'm telling this to you. So next week, I'm a graduate student from IASC. So next week, IASC is organizing this open day where the name itself says pretty much an open day. So everyone from all the other parts of India as well as from Bangalore are invited to come to the campus. So all the departments present here, we will be explaining our work. We'll be having demonstrations as well as some experiments regarding the work that we do in IASC. So do come here and do enjoy yourself around this uh, scientific open day. And since it is happening on Saturday, 1st March, which will be clashing with the regular TA session, so I have asked NPTEL team to postpone it to 2nd March, which is Sunday, from 3 p.m. to 5 p.m. Yeah. So the next TA session will be from 2nd March, which is on a Sunday, from 3 p.m. to 5 p.m. The other TA sessions from week 7 and week 8 will be on Saturdays only, hopefully, if there are no changes in the final schedule. But this TA session will be on 2nd March. With that being said, I'll keep this doubts room open till like for another 10-15 minutes. So in the meantime, you can ask any question, any doubts that you have in the meantime. So if you have any doubts, you can. There is no one in this meeting room here, but if anyone joins, you can post their doubts in the chat box. Or if you are watching this recorded lecture in YouTube, you can also post it in the YouTube comment section about the doubts that you have. Okay, if there are no doubts, I'll see you guys on 2nd March, next week Sunday at 3 p.m. to 5 p.m. Thank you.